Welcome to Cell Clips. In this video, we'll be discussing the transport of proteins through the mitochondria, the organelle responsible for producing energy used by the cell. If you saw our video on the zip code hypothesis, you know that after translation, proteins either remain in the cytoplasm or get transported to the nucleus, the ER, or the mitochondria. Before we dig into mitochondrial transport, let's build a map of the mitochondria. The mitochondria consists of two concentric membranes which separate each compartment. The outer membrane forms a protective barrier around the intermembrane space, and the inner membrane surrounds the matrix. This structure is essential for converting glucose into ATP, a usable form of chemical energy. ATP synthesis starts in the cytoplasm, where glucose is converted into pyruvate and then passed through both membranes of the mitochondria into the matrix, where enzymes catalyze the reactions of the citric acid cycle. The products of the citric acid cycle then donate electrons to a chain of enzymes in the inner membrane. This flow of electrons provides the energy needed to pump protons into the intermembrane space, creating the electrochemical gradient that stimulates ATP synthase and ultimately produces ATP. As you can see, this process requires many different enzymes, and these proteins must be placed in specific compartments of the mitochondria for these reactions to work properly. So, where do these enzymes come from, and how are they transported to the various sites where they are needed? Most mitochondrial proteins are transcribed from nuclear DNA and translated in the cytoplasm. Immediately after translation, these proteins are bound by chaperones to prevent them from folding. Next, they need to be imported into the mitochondria. This requires the help of translocons, protein complexes that mediate transport of polypeptides across membranes. Let's walk through what this transport process looks like. Proteins destined for the mitochondria contain an amphiphilic alpha helix at the N-terminus of the protein, called a mitochondrial import sequence. This zip code binds to a translocon in the outer membrane of the mitochondria, called the Tom complex. Chaperone proteins are then removed using energy from ATP hydrolysis, and the protein is pulled through the pore in the Tom complex. Once inside the intermembrane space, the rest of the protein's journey depends on its intended location. If the protein is needed in the intermembrane space, then it will fold and remain right where it is. Proteins destined for other parts of the mitochondria are again bound by chaperones. Those that need to be embedded in the outer membrane are inserted by another translocon called SAM. Proteins to be embedded in the inner membrane, including those in the electron transport chain, are inserted by TIM22. Proteins needed in the matrix bind a translocon called TIM23 that begins pulling the peptide through the inner membrane. As soon as a small part of the protein emerges in the matrix, a chaperone binds to prevent it from sliding back. ATP hydrolysis then causes a conformational change that simultaneously releases the chaperone and pulls the peptide further into the matrix. Then another chaperone binds, and this process repeats until the whole polypeptide is pulled into the matrix. This kind of transport system is called a ratchet. So far, we've talked about bringing proteins from the cytoplasm to the mitochondria. But now let's switch gears and talk about the proteins made from the mitochondrial genome. Because of its prokaryotic ancestry, the mitochondria has its own circular DNA that resembles a bacterial genome. It also has its own ribosomes, which synthesize proteins directly in the matrix. Some of these proteins remain in the matrix, while others are needed in the inner membrane. These proteins are inserted in the inner membrane with the help of the OXA translocon. So, to review, proteins in the mitochondria come from two sources: the nuclear genome and the mitochondrial genome. All proteins translated in the cytoplasm are transported through the Tom complex, regardless of their final destination in the mitochondria. Some proteins remain in the intermembrane space. Some are inserted into the outer membrane by SAM, or into the inner membrane by TIM22, and some are brought into the matrix with the help of TIM23. Proteins translated in the matrix of the mitochondria either remain in the matrix or get inserted into the inner membrane by OXA. Together, these pathways create the machinery needed for maintaining mitochondrial structure and function. You may have noticed that even while discussing the transport of proteins into the mitochondria, we've mentioned several processes that rely on energy from ATP. These are just a few examples that show how the energy produced by the mitochondria is essential for sustaining cell life. Well, that's it for this video. Thanks for watching Cell Clips.